Well, good afternoon. Today, Guillaume and I are very delighted to present you the final report on our project, Bridge. You have here the dream team with people originating from 7 different countries. All these people are plant ecologists. They are also specialists of plant or biological invasions, and some of them are also specialists of biological conservation and ecosystem restoration. The Bridge project was co-funded by ITECOP National French Project and FRB-Cesab. Its main objective was to understand how river corridors, roadside corridors, and field margins can modulate taxonomic and functional plant diversity within agricultural landscapes. On the photograph here, you can see that this kind of structure of landscape units can highly interact and can be close one to the others. At the regional scale, these three main linear ecosystems expand a huge importance as well in Europe as at the worldwide scale. They are very common and frequent habitats. Another important point is to point out the fact that some corridors can intersect one with the others. Here you have the example of a small watershed in the southwestern part of France, and you can see here the map of the river network of this watershed and also the road network. And if we consider the intersection between these two maps, you can see that bridges occupy all the regions and express a large set of elements that are very important for biodiversity. These three landscape units have some similarities, especially their configuration in the landscape, the fact that they are exposed to human management directly or through the environment. They have also, I just said that, numerous connections between these linear ecosystems, and when they don't intersect, they are often very close one to the others. So some first hypotheses. The first one would be that we expect a functional similarity or convergence between the communities hosted by these units. Also, as they are linear systems, we expect the fact that invasive species can be shared by these three landscape units. There are also some differences. The first one is the configuration, the spatial configuration of these units. You can see that river and road corridors are continuous systems in the landscape, while field margins are rather fragmented interfaces. And these habitats have also internal differences in terms of habitat diversity, uh, of habitat structuration, and of course of environmental variables that shape the diversity within each of them. So another set of hypotheses concerning these differences. We expect a huge effect of habitat filtering and a response to disturbance regimes that can explain the differences observed in biodiversity. We expect also more invasive species along corridors and along field margins. And we expect that uh, invasive and threatened species composition would be very specific to each type of habitat. The first task of our project was to build a database. This database includes species, of course, locations, of course, but also environmental variables and biological traits. So we constructed this database from 4 distinct data sets. These data sets were organized from south to north in the western part of Europe. And in addition to the location sites, we also included in the study the transverse gradients within each part of the landscape units, distinguishing an inner edge, an outer edge, and an interior. So here you have an illustration about where we sampled exactly within each landscape unit. As the construction of the database took a while, we decided first to focus on a particular data set in order to understand the effect of bridges on plant diversity. At the regional scale, we considered the succession of bridges along the river, and at an intermediate scale, I mean at the scale of a bridge, a given bridge, we considered sites located at the immediate vicinity of the bridge itself, and we compared what happened at the bridge with what could happen far away from the bridge as well along the road or along the river. And finally, at a very local scale, we consider the transverse gradient I've just described before. To analyze the data set, we, among other techniques, we used especially spatial explicit models in order to define the respective part of oriented processes and non-oriented processes, processes that could be involved, of course, in biodiversity dynamics. The questions were the following. The first question, do intersections alter or disrupt biodiversity continuity along road and river corridors? Second question, are biodiversity patterns mainly driven by directional or by non-directional mechanisms? And lastly, how much are local factors also important for shaping the communities at bridge locations? Well, today I will just give you some main results. I invite you to have a look to our paper if you want further details. The first observation we made is that rivers and roads shared a lot of species, as you can see. And when we compare the bridges to external references, you can see that as well external references and bridges have their own composition, their own species pool. A first set of results, surprisingly, we found that local species diversity was higher along roads than along rivers. We didn't expect that at all. Second point, it was less surprising. We found a reduced alpha and beta diversity around bridges. And also, we highlighted functional convergence of roadside and riparian vegetation at bridges. We found also that finally, directional processes controlling biodiversity was less important than diffuse processes across the landscape. This is a little bit counterintuitive since, especially along the rivers, biodiversity could be widely controlled by upstream to downstream processes. At the local scale, we found, as expected, a strong impact of local disturbance on biodiversity. We found also that diversity at the external edges is less original than in the inner parts of the corridors or of the interfaces. And here we can see probably the direct influence of the surrounding landscape. Surprisingly also, we found no differences in trait between invasive and non-invasive species. To conclude this first study, we can say that corridor intersection should be not overlooked for management purposes. The fact that bridges change biodiversity locally encourage us to recommend to managers to road and river managers to work together. Another point important also is that anthropized habitats like road corridors may correspond to suitable surrogate habitats for some threatened or infrequent species. And considering conservation strategy based on frame belt, green and blue belt considerations, we consider that non-corridor specific processes can also be taken into account for biodiversity dynamics and then for managing this biodiversity at the regional scale. Well, now I will unshare my screen and I will hand over to Guillaume to continue the presentation. Thank you.
Yes, thank you, Eric.
Maybe I have not.
Can you see my screen?
Yes, we can. We just have to put in full screen. That would be perfect.
Okay.
Yeah, perfect.
Okay. So I will present you the second part of the project. So in this part of the project, we wanted to compare the species pool of the three ecosystems. As Eric said, they have certain characteristics in common, for example, landscape configuration, human management. So we can expect a certain functional convergence. But these three ecosystems also have their own ecology and disturbance regimes. So we ask three questions. First, to what extent do the species pools of these three habitats overlap taxonomically? How do the functional traits of species pools differ across habitats in terms of ecological strategy, resource requirements, and responses to disturbance? And finally, what are the implications of this pattern for biodiversity conservation, especially for endangered species, and also for management of invasive species in linear habitats? So in order to compare these three ecosystems, we use the data that we pulled from all the partners of the Bridge project. So data coming from France, Spain, and UK. So as you can see on this table on the top right, the number of surveys per ecosystem is fairly well balanced but there was an imbalance by country with a deficit of a survey in Spain. So there was several options. We could have removed Spain, but it will have been a pity for the partner from Spain. So we tried to keep them by resampling the data to balance the sampling effort between habitat and region. So we used a stratified resampling procedure with the probability of drawing a survey inversely proportional to the number of survey in a given region. So after this resampling, we obtain an average frequency for each species in each habitat. And on this basis, we carried out a taxonomic and a functional comparison of the three ecosystems. So at the taxonomic level, first we compare the gamma diversity. So that is the total diversity of the species pool of each ecosystem. Then we were more interested in the taxonomic similarity and the number of species that are specific or shared between the three ecosystems. So for that, we used a Venn diagram, as you can see on the slide. So we can define 7 groups of species, 7 kind of species pools. So you have 3 groups that are specific to each habitat, so G1, G2, G3. You have a group of species that are shared between 2 of the 3 habitats, so G5, 6, and 7. And finally, you have the most generalist species, G4, that are shared between all the 3 ecosystems. So this analysis, you can do it with all the dataset, so using the raw dataset. But in this case, you will have some species that are present in an habitat just by chance. That's why we also applied the INVALD procedure in order to identify species that are statistically associated to each habitat or significantly share between 2 habitats. Then in a second step, we also compare the 3 ecosystems based on a functional approach, using also two stages. The first stage was just the comparison of the mean trait distribution across the 7 groups. And in a second step, we estimated the functional richness and dispersion of the 3 main species pool using an hyper-volume approach. So let's see some results now. So on this slide, you can see the richness accumulation curves for the 3 ecosystems. You can see that riparian and roadside ecosystem have a much higher gamma diversity than field margins. So if you took a resampling of 3,000 records, you can see that for riparian and roadside, you have about 1,500 species and only 1,100 species for field margins. So this means that probably they are more constrained in field margins. Not all species are able to cope with the conditions and filtering in these habitats. So then we look more into detail in the number of species specific and share between the 3 habitats, sorry. So on the left, you can see a Venn diagram with a number corresponding to all the database. So it's a raw comparison, including rare species, as well as species that could be considered casual in each habitat. And on the right, you have the result with the INVALD analysis, which filters out casual species. And in this part also, we excluded species that were less frequent than 1% in the sample of each kind of habitat. So what can we say about these 2 figures? Um, there are several points in common. So the first is that you can see that riparian habitats host the highest number of specialists. So it's probably the ecosystem with the most specific species not found in the two other habitats. Second, we can also see that many species are shared between the three habitats. So it was one of our first hypotheses. It is about a third of the species if you take the raw comparison, and a fifth of the species with the indicator analysis. And the last point, you can see also that the two habitats with the greatest number of characteristic species in common are field margins and roadsides. So this is consistent because if you look at the location of field margin in the landscape, they are often very close to roadsides. Sometimes it's even the same habitats, and they are also managed in a very similar way with mowing. So the second step of this analysis was to do the same, but this time we focused on particular species. So on the left, we look at invasive species, and on the right, you can see endangered species. So in our data set, we have 68 invasive species and 47 species that are listed on a red list in France, Spain, or UK. So it's very interesting. If you look at the two figures, you can see a completely opposite pattern between these two group of species. On the left, you can see that invasive species are mostly shared among the three habitats, meaning they are primarily generalist species able to cope with very different ecological conditions and disturbance regimes. While in contrast, endangered species are mainly specialists of the three habitats. A second interesting point is that invasive species are also widely shared between riverbanks and roadsides. And this could illustrate the corridor role of these two habitats, which favors the spread of invasive species. And this pattern is less evident along field margins, which are more fragmented. A third interesting point is that also a river has the most specific flora. You can see that roadsides and field margins should not be overlooked in terms of endangered species because they host as many endangered species as river banks. And this may be explained by the refuge role of these linear habitats. When the adjacent patch, for example, a cultivated field, is managed more intensively compared to the margins. So in relation to this, you have also on this slide the frequency of occurrence of invasive species in red and endangered species in blue. Note that the scale is logarithmic. So this was just to show first that even the invasive species are not so frequent in our data set. The most frequent invasive species have a frequency between 10 and 50% of the sample. And you can see that these invasive species are more frequent on river sides compared to field margin and roadsides, while endangered species are slightly more frequent in field margin and roadsides. So we've just seen that there are taxonomic differences between these three ecosystems, but what about functional differences? So on this slide, you can see a few graphs comparing the distribution of trait values. So specifically for SLA, specific leaf area, where you can see that SLA is higher in field margin on the left compared to road in green, with the riverside having intermediate value. You can see also the Ellenberg indicator value for soil moisture. And of course, here you have higher value for species of riverside. Secondly, for field margin and roadsides have more drought tolerant species. So we did that for a number of traits, including seed mass, plant height, other Ellenberg indicator, and also disturbance indicator. And in summary, we found some differences in trait value, but more marked differences in ecology and disturbance regimes. So in summary, in field margin, you have more annual species with high SLA value, which is very consistent with this more productive habitat, very frequently disturbed by farming practices. And so the species have also a high indicator value for the frequency and the severity of disturbances. On roadsides, the species differ from the two other habitats by being more drought tolerant and preferring full sunlight. And in riparian zone, you have a species requiring higher moisture, also more geophytes, small seeded species, and water dispersed species. So these are about the mean condition reflected by the species. But what is also interesting is to see on the box plot, you can see, for example, that the range is very higher for river compared to field and roads. And this is what we have investigated when we analyze the functional diversity of this three species pool. So one difficulty to assess the functional diversity is that it can appear artificially higher simply because the pool contains more species. So that's why we based our calculation on the number of species in the smallest pool of species, which was field margin species pool, with 66 species specific to field margin. And then we computed 1,000 hypervolume for riverbanks and roadsides by randomly drawing 66 species from each of their respective pool. So the advantage of using hypervolume compared to other metrics for computing functional diversity is that it can better account for complex multivariate space, non-convex shapes, and so on. So here you have just two results. The first one is correspond to the functional richness. So it's the volume of the hypervolume on the left, and on the right, it's the functional dispersion. So it's the average distance between a sample of species in the hypervolume to the centroid of the hypervolume. So it's more about the even distribution of species within the hypervolume. So what you can see on the graph is the distribution of the 1,000 values of the hypervolume for river and road, and the unique value for field margin. So we also display the 95% of the distribution of river and road. And you can easily see that the species pool from the river have a much higher size of the hypervolume, higher functional richness, while it's not different between field margin and roadsides. And for the functional dispersion, again, the river has the highest functional dispersion, but this time the species pool from the roadside was higher than that of field margin. So this suggests that for a similar volume between field margin and roadsides, species in field margin are probably more functionally convergent in a part of the hypervolume with a few outliers. And the more general conclusion of this part is that with a higher functional diversity for the riverside species pool, probably it can be explained by 2 points. The first is that riversides have probably a greater habitat heterogeneity, offering a wide range of ecological niches for species. there is a strong gradient from close to the river and higher elevation and more dry part of the river corridor, and also weaker and less predictable human disturbances, which allow for the persistence of more functionally diverse species, which are not filtered by more regular management practices. So this is part of a manuscript that is currently in preparation and which we hope to submit by the summer. And if we combine the result from the taxonomic and the functional approach, you can see that Riverside is the taxonomic richest, also the richest in terms of functional diversity, and it has also the most specific species. So it is probably the most important corridor, but in the same time, field margin and roadsides have also should not be overlooked in terms of conservation. We have seen that they have also some specific endangered species. So it's also important to keep that in mind. So during the bridge project, we also developed a common project with Navidiv, which was another CESAB group, which was about the impact of navigation and navigated waterways on biodiversity. And so we worked together on an opinion paper about the impact of all transportation linear infrastructure, including roads, railway, and navigated waterway. So our idea was that these linear infrastructure are common and will keep growing in the coming decades. So it's important to better understand how to reduce their negative environmental effect and find places where they might help. And we suggest in this article that linear infrastructure could help species move in response to climate change. So because we know the limitation on species' ability to shift their range to keep track with climate change, management strategy at the landscape scale should be developed to facilitate species dispersal and climate migration. So this includes the management of existing linear infrastructure units and the design of new one to provide a valuable opportunity for land manager and stakeholder to explore if and how this infrastructure could facilitate plant and animal climate migration in the coming decades. Okay, so this led us to the management implication of the bridge project. So we have seen that the three linear ecosystem, field margin, roadsides, and riparian zone, they all play a vital role in enhancing overall landscape diversity. They also act as ecological corridors, which can have both beneficial but also negative impacts on biodiversity. And among them, clearly the riparian ecosystem is the most diverse and ecologically specialized habitat. Roadsides and riversides both serve as corridor, and we have seen they tend to facilitate the spread of invasive species more than field margins. And importantly, also, field margin and roadside should not be overlooked in landscape management, particularly for the conservation of endangered species. And we have also to think about interaction among these corridors, which could contribute to increased habitat diversity and landscape heterogeneity. And finally, an important aspect also is to try to preserve sources of rare specialist species and maintaining connectivity to support the recolonization of restored habitat. Okay, and just to finish, some words next to science about our feeling and maybe our advice after having coordinating a synthesis group at CESAB. So maybe the first point is that working outside of the lab, even through video conference, has actually made our workshop more productive. It has been a really positive experience for both the PIs, but also for all our partners. We would like also to say that the facilities at CESAB are very excellent, thanks to the CESAB team for their hosting. And what we like is that researchers can focus fully on their work without worrying about administrative tasks. So this is a big help. And the infrastructure and the human support at CESAB have been especially beneficial for the postdoc. Another plus has been the opportunity to meet other people and other groups, like in our case, Navidiv, which fosters collaboration and networking. And of course, we like also the technical support from CESAB, the data scientist. For example, with Nicolas Casajous, we developed an R package to solve a problem we had in analyzing the data for the first paper. So this was very high value to our work. Of course, we also face some challenges. First, it has been hard to get all partners together in person for plenary workshops, especially because in our group we had two people from America. It was a bit more difficult. And maybe also we didn't have enough time at the beginning to really work on constructing a single database. But maybe because at that time we were not aware that data scientists at CESAB could also help on this part to propose their help to clean the species list and gather data on traits. So my advice will be to really discuss as much as possible with data scientists. They often have the solution to our problem. Okay, that's all for me. Thank you very much.
Well, thank you very much to both of you for this nice presentation. Now I think it's nice if you maybe unshare your screen so that we can see you and also the people that would have questions. So yeah, I see that we have some questions in the chat that I don't know if Antoine wants to ask them orally, or if you're not able to talk, I can also read them out. Otherwise, if people want to ask questions, please just raise your hands. Oh, and I forgot to take my video on. Okay, so just please raise your hands and I will distribute the questions. So Antoine, yes, I will read your questions then. Thank you very much for posting them. So his first questions are, what kind of rivers are there in the study? Because size and type of bridge could influence the results. I don't know if, Eric or Guillaume, you want to answer?
Yes, I will try to answer to this question. Actually, we use a wide range of river size. Rivers range from 1 to 6 order size. That means that we started with very small streams, and the study gathered also very large rivers. Actually, this is a real problem because, of course, these different sizes of streams imply that the vegetation would and the ecosystem would work very differently from small to big rivers. But in our analysis, we control for this parameter. Especially when we worked along some rivers, we controlled for the river size and the river location along the river continuum.
Right. Well, thanks for that answer.
Yes, of course. Yes, I see the next question. Of course, yes, there are a large difference. We observe differences according to the size of the river. But it's a little bit difficult to decipher the effect of the size by itself because, of course, for example, small rivers are located rather in mountainous regions, at least in the South Europe, and so there is also an effect of the climate and we could difficultly we couldn't disentangle between the effect of the climate and the effect of the river by itself. But for example, for invasive species, there are no significant differences in terms of proportion of invasive species between small and large rivers. Of course, the species are very different.
And okay, thank you. And there was another question from Antoine in the chat where he was talking about the effect of wood inside the stream. So on natural
Yeah.
rivers with a lot of wood and beaver, the result should be much higher. The invasive species should also reduce a lot with this wood diversity and restore natural habitat.
Well, we didn't target specifically wood gems, for example, within the river corridors, but of course, it is very important because woods are involved. Dead wood is involved of course, in nutrient recycling, but also in river geomorphology. And so they are probably important, but the problem is that from a country to another, the way that wood gems are treated is very different. For example, in France, the wood gems in the river courses are removed by law. The law obligates managers to remove the dead wood from the main course of the river. And so in some other countries, they don't do that. And so it's difficult to include, but we haven't specifically worked on this problem.
Okay, we have a question from Fabien. So Fabien, if you want to unmute yourself and yeah, thank you.
Yes. Hi, everybody, and thank you very much for
Hi.
this fantastic talk about the Bridge project. I really enjoyed it. And I was wondering regarding the first part of the project and the bridge experiment with the control and the sampling at the bridge. You have this distance between the bridge treatment and the control treatment. And I don't remember if you mentioned what is the physical distance between the samples of the bridge and the samples of the background, and
Yeah.
how did you calibrate that?
Well, it was a little bit complicated because actually it depends on the distance separating two consecutive bridges. And so this would be the middle or between distance between medium distance between two bridges would be the best way to define that. And when we have it's about I would say that reference sites were both along the roads and along the rivers were about 3 kilometers from the bridge itself.
Okay, thank you very much for this precision.
Thanks for your question. There's another question in the chat from Savannah. So question about the indicator species. If you could repeat which analysis this was. If there's a
Hi,
specific
Savannah.
slide. Yeah.
Yes, it was the Inval analysis, which was introduced by Legendre in 1996, and there was several new versions. You can easily find this in several R packages to compute it. So it selects really the species that have a frequency and an abundance that is significantly higher in one of your group compared to the other groups, for example. So it is widely used in ecological analysis to find out specific species of cluster analysis and so on.
Okay,
Yes, I think
Lisa.
lab
Oh,
DSV
sorry.
is okay. Yes.
Hello. I was wondering, because your analysis focuses on plant diversity, do you have an idea if this would be, if you would have comparable results with other taxa, or do you expect it to be very different, or what are your thoughts?
Maybe I will answer, but well, first there is for some groups there is a direct relationship or a direct dependency between animals, for example, and plants. So, for example, for pollination or processes like that, we can make the link. But clearly, well, I'm not really, I'm no longer a specialist of animals. So I would say that we cannot transpose our results to other groups like microorganisms or animals. It's very plant specific, I think. I don't know what Guillaume thinks about that, but this is my feeling.
Yes, I agree. Maybe for the corridor role of river and road, maybe we can find some similar results with at least some animals. Maybe not all, but yes, in general, maybe our results are more plant specific than.
And maybe for aquatic animals, I encourage you to have a look at the report of the Navigative project. They have worked specifically on this aspect. Even they are focused on navigable waterways. There are some indication on how fishes or macroinvertebrates could behave along this kind of um, of aquatic corridors. But for other corridors, we have, for example, we have very few information worldwide about the way that insects can move or colonize the roadsides, for example. So there is some lacks of information for some groups.
Thank you.
Thanks, Lisa, for your question. I have a question as well,
Mhm.
maybe. You were talking about the differences in connectivity between the three types of habitats, right? Of structures. So there's the rivers that are continuous, the roads that are continuous, but the field borders that aren't. Is there a way, or would you think about a way of specifically testing the effect of the connectivity, especially since you were also opening the questions about the role of dispersal of species?
Mhm. Yes, another important difference between road networks and river networks is that uh, river networks are built according to a hierarchical geometry, while roads are rather meshed networks. There is no hierarchy in these networks, and this is a very important question, but we didn't solve that because first our samples were too much scattered or too much focused on a single upstream to downstream continuum, for example. But a way to analyze that, we did it partially, is to use spatially explicit models.
Yeah,
We
yeah.
use we use the eigenvector vector maps in this purpose just to see how much directional or non-directional processes were involved or not. But with this kind of model, we cannot identify which process exactly is involved. We know that they are directional or not directional, but we cannot identify that. And maybe another track to solve this problem is to go to the DNA analysis on some particular populations. Of course, we cannot do that on thousands populations, but we can track genetic continuity and diversity along the networks in order to see how much effectively plants, for example, plants living on the road can migrate to the river corridor or conversely.
Okay,
I don't know.
interesting. There is a question in the chat, but I think Guillaume just answered straight by reading. I don't know. Is there anything else you would want to add?
Yes, I answered maybe only partly to this
Yeah,
question.
maybe read the question again since I didn't do it. Yeah. So the question was about characterizing the roadsides. So whether the ditches and hedges were taken into account.
So maybe Eric, you can develop further my answer because you are the roadside.
Yes, well, I would say that comparatively to riverside, roadside were quite easy to sample because they are mainly constituted by strip-like habitats. So we could easily distinguish between a first strip very close to the pavement, which is dry, hot, exposed to sunlight. Of course, we can identify also the external edge of the road corridor, but the more difficult was to identify the interior because between the inner and the outer edge of the corridor, it could happen a lot of things, just like along rivers. You can, for example, you can find a ditch along the river or not. So you can have wetlands along the river roads, and you can also have very dry habitats. We use particular, from a very practical point of view, we use very particular size of quadrats. We used 0.5 by 2 meters long quadrats, so they were rectangular quadrats to sample vegetation because we wanted to favor the longitudinal sampling comparatively to a transverse analysis. But we did also, as I explained in my presentation, we did also a sampling according to transects from the interior to the exterior of the road corridor. I don't know if I have answer to the questions.
No, I think so.
Concerning the integration of the landscape, this is another story we are working on with Lindsay Maskell, who is our British partner. So she's working on the comparative effect of the landscape configuration and the local configuration of the habitats and the effect of these two scales on the community structure.
Awesome. So maybe we can take one last question from Fabien, and then we might call this a session.
Yes, thank you. It's a bit follow-up on my previous question. It's I was quite stricken by the number of species in the bridge experiment that are only found at bridges, which is quite important. And I was wondering whether you were able to distinguish between what is due to sampling stochasticity, meaning that every time you sample, you can find new species that were not in the previous sample, but just because of the sampling, and what is due to maybe local practices at bridges or a microenvironment effect at bridges, and that would explain a community differentiation at the bridge precisely. And especially, I'm thinking about whether you can compare the two samples that are far away from bridges, but at both sides, whether they are more or less different than the bridge at the middle, or whether you try that kind of thing. That's an open question.
Well, we control for that. Clearly, the fact that well, there is two big influences of for the biodiversity within our bridges. The first one is the location of the bridges themselves at the regional scale. So they are influenced by climate, soil, and so on. And the second one, and we could highlight it very well. The second reason for which the flora or the vegetation at bridges are very different is linked to the way that bridges are managed, especially by mowing or changing the morphology, the geomorphology around the bridges. So it is a clear effect of managing practices that can also modify the flora and make this flora more original than the surroundings.
Okay, thank you.
Awesome. Well, thank you, everybody. Thank you, Eric. Thank you, Guillaume, for this nice presentation and for all the love to the CESAB. We love it. It was good to have you. And thanks, everybody in the audience, for being here and asking nice questions. And so, yeah, I'm now going to stop the recording and we will put it on our website where you can also find further information about the upcoming Cesabinars once we will post it. And also rewatch the previous sessions if you want to. Thank you.